皆様、弊社住商ファーマインターナショナルのランチョにお越しいただきましてありがとうございます。お時間になりましたので始めさせていただきたいと思います。私どもですね、今回のランチョのテーマであるイノクラスによる全ゲノム解析サービスのご紹介をさせていただきます。イノクラスのサービスは昨年の11月より始めておりまして、西原先生にはですね、一番最初の早い段階ではトライアルをしていただきまして、今回ですね、ゲノム医療の新たなページにおけるバイオバンカーの意義を考えるという、第10回のこの非常に記念すべき回で、西原先生をお招きしてランチをさせていただけることを大変光栄に思っております。西原先生誠にありがとうございます。イノクラスですけども、2020年に設立しましたアメリカの企業でございまして、午前中のセッションでもございましたけども、全ゲノムの課題である新鮮凍結サンプルでなくてもですね、FFT からでも全ゲノム解析をできるというところが特徴でございます。またですね、全ゲノム×全ゲノムということで、全てトータルを全ゲノムでやる MRD の交換度システムを持っておりまして、そちらも今日ご紹介をさせていただく予定でございます。本日ですね、演者2人、お招きしておりまして、1人目はですね、チャンソクリー、英語名でジャスティンと言います。どうぞですね、懇親会にも来ますので、気軽に話しかけていただければと思います。ジャスティンですけども、ソウル大学の医学博士号を取られまして、その後ですね、カイストでヒト免疫学の博士号を取得している MD-PhD にございます。現在ですね、イノクラスの最高イノベーション責任者として、外部のイノベーションのパートナーシップおよびですね、韓国事業のゼネラルマネージャーを務めておられます。Justin, stage is yours. Please get this started.
Introduction real quick. Okay. こんにちは。英語で話すことをお許しください。I've never wanted to speak Japanese like this today, so I really wanted to learn it. Actually, my first daughter's major is Japanese language in high school. I'm very happy to meet you guys. So good afternoon. It is a great honor to present our company's whole genome-based cancer genomics analysis and diagnostics to such distinguished experts and academic leaders in Japan. So I'm Jeong Lee, scientific founder of Inocras. Today, I'd like to briefly introduce how we are transforming oncology with whole genome precision. Our approach is built on three pillars. First, we are using whole genome cancer profiling, offering a comprehensive view of tumor genomes to guide personalized treatment. Second, MRD, redefined using whole genome to whole genome comparison for ultra-sensitive detection of residual diseases. And finally, we are trying to provide confidence to advance, generating real-world evidence that benchmarks whole genome against currently best clinical standards. Through this, we aim to enable more accurate and data-driven cancer care in the real world. So let me briefly introduce Inocras. We are a global genomics company based in San Diego, California. Actually, we started our company in Korea, but we moved our headquarters to California about 4 years ago. Our mission is to take large-scale whole genome data, traditionally used only in research, and make it truly useful for patients, healthcare providers, and drug developers. So far, we already have analyzed data from over 15,000 patients with cancer and rare diseases using our own whole genome sequencing platform. All of our clinical testing is now performed on the CLIA, CAP, and ISO certified standard. Currently, we offer two flagship products. First is Cancer Vision. Our tissue-based solution provides one of the most comprehensive analysis of somatic mutations in cancer. And second, MRD vision. This is liquid profiling platform, achieves ultra-sensitive detection of minimal residual disease with a limit of detection down to 10 to the minus 6, offering one of the most sensitive MRD assays available at this moment. So why do we need whole genome sequencing? I also joined the conference in the morning, and I know Japanese scientists and leading doctors are now discussing very actively about whole genome sequencing these days. Here, you can see two surface plots comparing mutation profiles in cancer genomes. On the left, we have what's typically captured in current targeted panel sequencing. So only select biomarkers, and it is very limited detection of point mutations, copy number variations, structural variations, and tumor purity estimates. In contrast, whole genome sequencing offers a truly comprehensive view, more mutations identified, of course, and more accurate genome-wide copy number variations and structural variations, coverage of non-coding regions, and also for precise genomic markers like tumor mutation burden, microsatellite instability, and also for homologous recombination deficiency. And this can be related to better tumor purity and probability estimates also. And even we also include mutational signatures. This is why more and more clinicians and researchers are choosing whole genome, because it sees what panels miss. So Cancer Vision compares the whole genome of FFPE tumor tissue with matched blood. Results can be delivered as a paper report in here, and also through our interactive genome viewer with optional support from molecular tumor board based on Zoom or Google Meet based not in-person meeting. And also, Cancer Vision is now actively providing patient services for clinical trial matching and also for genetic counseling in US. The entire process is ClearCap certified and completed within 2 weeks, as fast as or even faster than most advanced panel tests. And Cancer Vision is not just about the scale of whole genome data. It's built with two key features that address real-world challenges faced by clinicians and researchers. Let me briefly introduce about our two technical features of Cancer Vision platform. First, we offer target-enhanced whole genome sequencing. This means you can get both whole genome coverage and deep sequencing of key tumor mutation regions. So combining these two insights, we can deliver the whole genome sequencing with the reliability of a panel. And of course, a germline analysis is also included. So that means it's a kind of 3-in-1 test. We give a 500x panel, and also we include 40x whole genome using FFPE tissue, and also for germline test result. And while I'm not here to talk about pricing actually, but I will just say this. We provide all of this at a comparable cost to traditional panels all over the world, including in Korea. So that's why we can adopt this Cancer Vision platform in Korea earlier, and it's already reimbursed by national insurance system, and currently on the Medicare review in the United States. Second technical key feature of Cancer Vision is FFPE correction algorithm. Unlike most research-grade whole genome platforms, we achieve highly reliable performance even from FFPE tumor samples. This is made possible by our machine learning-based in silico filter technology, which corrects formalin-induced damage during the tissue fixation process. The filter has been iteratively improved and validated using data from over 10,000 clinical cases already, and making it one of our core proprietary technologies, a unique IP that sets us apart and cannot be easily replicated. Cancer vision has been rigorously evaluated and published in peer-reviewed literature. In a head-to-head comparison with one of the most widely used target panels, we successfully detected all somatic mutations reported by panels without exception. But more importantly and unexpectedly, we found that traditional panels which analyze only tumor tissue often misclassify germline mutations as somatic, because cancer vision basically analyzes both tumor and matched blood. It avoids this critical error, highlighting another key advantage of our approach. In this prospective observational study, the primary objective was to assess the clinical utility of implementing whole genome sequencing into a real-world hospital setting. In category 1.1, which involved selecting targeted therapeutics, this is the red part in this graph, the utility was around 30%, which is comparable to that of conventional panels. However, category 1.2 and category 1.3 and category 2, facilitating clinical trial screening, eliminating ineffective treatments, and providing clinical clarity was the additional value of cancer vision test. Cancer vision demonstrated a substantial additional utility, reaching the overall clinical impact to nearly 70%, which is almost twice times more than usual panel clinical utility. In other words, at the same cost and turnaround time as when we compared with conventional panel, we deliver 3-in-1 test that provided twice the clinical value. There is another urgent reason why whole genome sequencing is not just valuable in academic area. Now, it is very clinically essential test. It's not only about depth or breadth of data, but about avoiding critical misclassification that directly impact patient care. For example, in HLD assessment across more than 1,000 breast cancer patients in Korea, cancer vision clearly separated positive and negative cases, while standard panels show only high false positive rates due to arbitrary cutoff here and limited coverage. That's why leading breast and gynecological oncologists in Korea nowadays rapidly turning to whole genome-based solutions as their genomic testing. The comprehensive tissue mutation profiling provided by cancer vision also serves as a powerful foundation for tumor-informed MRD testing. At Inocras, we also applied duplex whole genome sequencing technology to cfDNA, and it enables highly precise and ultra-sensitive detection. So more markers mean greater sensitivity in MRD testing. And with no need for panel customization process, the workflow is simpler and more cost-effective than usual panel-based MRD testing. This powerful whole genome-based MRD is now demonstrating its ability to detect relapse earlier with ppm, that means 10 to the minus 6 level sensitivity. This is one of the highest levels of precision in this field, and the usual panel-based MRD is currently guaranteed only 10 to the minus 4 level at this moment. So this is the most sensitive MRD in the world at this moment. We conducted a direct comparison between our MRD vision and the leading panel-based MRD test. In lung and ovarian cancer patients, MRD vision demonstrated strong agreement with the panel in here, while detecting residual disease at lower tumor levels. Unlike panel assays, MRD vision leverages whole genome sequencing to expand the detection and avoid the limitations of predefined targets. Simply put, MRD vision matches the accuracy of panels in the higher level of MRD, and while offering broader and deeper insight with the strong strength of cancer vision tissue profile. MRD vision uses whole genome for both tumor and cfDNA, enabling ultra-sensitive detection. It identifies low-level tumor DNA missed by orthogonal test here, and the MRD signal patterns over time were consistent, even in samples only detected by MRD vision. That means these two cases are very likely to be true MRD positive with a very lower level of detection. So importantly, all blank samples were confirmed as negative here, so supporting the test's high specificity too. So this slide highlights how MRD vision outperforms conventional MRD products across multiple dimensions. First, both tests are tumor-informed, of course, but MRD vision uses whole genome sequencing for both tumor and cfDNA, as I told you, while most existing assays rely on targeted panels for cfDNA detection. This allows us to achieve ultra-sensitive detection, down to 10 to the minus 6 level compared to typical 10 to the minus 4 panel. In terms of deliverables, we provide both cfDNA MRD report and a full tissue whole genome profiling altogether, giving clinicians a more comprehensive view. Thanks to our streamlined workflow using Ultima, this is a brand new sequencing platform from US. They provide a PPM6 platform, which is also a duplex sequencing platform. We can also offer this at a more affordable cost with a fast turnaround time, just 2 to 4 weeks for initial reports and 7 to 14 days for follow-up report. So in summary, MRD vision delivers greater sensitivity and richer insight. So this is the last slide, and as a physician scientist myself, my mission is to make this fast and transformative genome data accessible to everyone. So from my experience, especially with leaders like Dr. Nishihara, Japan is a place where some of the most thoughtful and rational physicians and scientists are making truly important decisions. We sincerely hope that our technology can be of help to you, and I sincerely appreciate your interest and support. Thank you.